Hello, everybody. My name is Magdalini Christodoulou, and I'm a researcher in Seckenbeck. I'm currently working in an Interreg project, GENES, which aims in developing a reliable DNA barcode reference library for the identification of benthic invertebrates, essential for biomonitoring of the North Sea. GENES is an acronym for Genetic Tools for Ecosystem Health Assessment in the North Sea region. You can find more information for this project in the link you see on your screens, and you can follow us in Twitter. GENES is European Union funded. It has a duration of 3 years. We are currently asking for an extension, a budget of 2.5 million. It has a consortium of 9 partners, which you can see on the bottom of the screen, and it's been led by ILVO in Belgium. The main objective of GENES is to implement DNA-based tools in routine monitoring programs in support of policy and decision-making concerning ecosystem health. Benthic macroinvertebrates are key components in environmental impact assessment, but their use as bioindicators can be constrained by the time and cost-consuming processes needed for their morphological identification. The recent advances in high-throughput sequencing, for example, DNA metabarcoding, can provide an alternative to morphology-based approaches. The main limitation is the considerable investment needed to build high-quality and curated taxonomic reference libraries for species identification. GENES, for this reason, aims in developing a curated DNA reference library based on mitochondrial CO1 gene, the barcode gene for the North Sea macrobenthos that will serve as the backbone of all the molecular protocols developed within GENES, and not only. In order to begin our reference libraries, the partners of GENES came together and we compiled a key list of 800 species, meaning that we aim to sequence these 800 species, of which 96 were niche species, non-indigenous species. These species were selected because they were found in the pilot studies that we were conducting within GENES. On your left, you can see the information on the species, in which phylum they belong, in which class, and the AFIA ID of the species based on worms. On the middle columns, you can see which partner has this species in its pilot and if it has a sequence or a specimen in its collections. And on the right columns, you can see whether this species has a sequence within the pre-existing GENES reference libraries in bold, in GenBank, and whether this species is invasive or not. In order to find our species that we were aiming for, we started a new sampling campaign, as fresh material is always easier to sequence. We also started scanning our collection, our voucher collections that each institute has, and we also checked our existing reference libraries, DNA reference libraries. For example, Seckenbeck and Mer has a lot of North Sea barcode projects. Here on your screen, you can see some of the examples, like for decapods, for crustacean larvae, various for North Sea and Baltic Sea fauna, and for ephemerits. We validated our data so we could later add them in the GENES reference database. We follow a standardized workflow, lab workflow, where we aim for the mitochondrial gene CO1, and specifically for the 658 base pair Folmer region. We extract our DNA, we amplify it using PCR. We sequence our specimens either in-house or in Macrogen, and then once we get our sequences, we process them through Genius, where we make our quality control. All the specimens that they have been sequenced are being also photographed at the same time. So this way we create a photograph library, a voucher library, and also a DNA library online in bold. We also follow a curation workflow that has to do mainly with the data deposit in bold, meaning that we make sure that all the specimens are identified, checked eventually by expert taxonomists, especially when we have a taxonomic problem. We make sure that all our specimens have a voucher deposit. We make sure that the accompanied specimen metadata are deposited in bold. We follow barcode standard, meaning that we do sequence quality controls. And if it's not up to our standards, we repeat the sequencing. We constantly update the taxonomy based on worm, especially the nomenclatory, or if we have new species. We cross-reference our data among institutes, meaning that when we update, for example, Ilvo and Seckenberg can upload sequences of the same, belonging to the same species. We make sure that these sequences are aligned and they do belong to the same species. We always keep track library versions. We tag our sequences with markers. For example, that the species has been validated, or it's contaminated or misidentified. And we always create our new entries in cross-reference with the old entries, and if needed, we revise our old entries. Here you can see an example of common hermit crab in the North Sea, Pagurus bernardus, and how we upload our data in bold. On your left, you can see the taxonomic information of the species accompanied by photos. You can see also that the species is being tagged as validated, meaning we have no taxonomic pending issues. Below, you can see all the collection data information that accompanies this species, and specifically this specimen. And on your right, you can see the sequence information along with the trace files. Here, I'm showing an example of a new species, Loimia ramseka, that it's a worm recently described. It's a niche species, and through BOLT and the genes reference database, we verified to the stakeholders that gave us the specimens that they have a new niche for the German seas. This species was confused with Lanitze conchilega, and it was very easy with the DNA barcoding to separate these two species and go back to the stakeholder with information regarding this niche species. As time progressed, after accumulating a lot of material, our key list expanded with specimens of species that we didn't have in the original key list. So we started being ambitious of having more species. So here in this list, you can see with green the species that we have a voucher, but we don't have a sequence yet. With no color, the species that we have sequence already. With orange, the species that it's an addition to the new key list. And with red, the ones that we don't have neither a species, neither a specimen. In this graph, you can see the progress of genes in relation to the key list that we have compiled, the 976 species that belong to 15 phyla. For example, here you can see that with light blue for the mollusks, is shown the species that we have sequenced. With dark blue, it shows the specimens that we have a voucher, but we haven't got a sequence. And with gray, shown the specimens that we have, the species that we have neither a sequence, neither a voucher. You can see that we are way ahead for mollusks and for arthropods. For the echinoderms, we are almost completed in relation to our list, while for the annelids, we still have a lot way to go. We wanted to see in the future where we stand in relation to the species found in the region. So we compile a list based on the literature, which species are found in the German waters, in the Dutch waters, and in the Belgian waters, because this is where mainly our sequences come from, as Naturalis, Seckenberg, and Ilvo are the main partners within genes that they're contributing with sequences. And we found out that 1,432 species have been reported from this region that belong to 16 phyla. Again, here you can see that for the number of, it's shown with light blue, the number of species we have a barcode, with dark blue, the one that we have a specimen, and with gray, the ones that we have neither species, neither a specimen. As you can see in this graph with the mollusks, we are quite advanced, almost completed. The same with echinoderms. We are way advanced in arthropods, but we are behind in annelids, bryozoans, porifera, and invertebrates. Currently, the genes reference library holds 3,507 specimens that belong to 592 species or in 613 bins. Within, they belong to 11 phyla, and for all these specimens, we have the same number of CO1 sequences, and for some, we have a few 16S and 18S. In a future work, we aim in acquiring more genes for these species. Here you can see an example for the first 200 species, an accumulation curve for morphological species and barcode index numbers. And you see that the number of bins is always slightly larger than the number of species. This is perhaps because cryptic species are awaiting discovery, or because the species intraspecific diversity is high. So, in conclusion, currently genes reference library holds 3,507 CO1 sequences that belong to 592 species, aka 613 bins. Once completed, the genes reference library will be the first well-curated DNA library covering all the spectrum of macrobenthic animals in the North Sea. And once publicly available, this DNA library will support the implementation of fast, cost-efficient, and reliable DNA-based identifications and subsequently environmental health assessments in the North Sea. Here, I should say that this is a group work, a collaborated work, and we need all the help we can get, and we are more than open to new collaborations in order to get new voucher specimens, sequences, and we will be happy to talk with anybody who is interested. Thank you very much.